Hi everyone. In this video, we're going to look at an overview of the process of gene expression, that is, using information in DNA to make functional proteins. Gene expression in eukaryotes starts in the nucleus with the process of transcription, the reading of DNA, and the synthesis of a complementary messenger, or mRNA molecule. Once this is made and processed, it needs to be exported through a nuclear pore to the ribosomes in the cytoplasm. Here, the process of translation can be performed using the sequence of bases in the RNA to dictate the synthesis of a polypeptide chain from amino acids. We'll look at the details of transcription more in another video, but for now, we're going to focus on how the information in each molecule is used. We start with a double-stranded DNA molecule, but we're only going to use one of these strands for transcription. The one that we're going to read is the template strand, and it's going to be read in the 3' to 5' direction, because just like DNA polymerase, RNA polymerase also synthesizes a strand that is anti-parallel to the template and does so in a 5' to 3' direction. We refer to the non-template strand as the coding strand because it has the same sequence as the RNA that will be produced, with the exception that T's in the DNA will be replaced with U's in the RNA. RNA polymerase will bind to the DNA and move down it, reading the template 3' to 5', synthesizing the complementary mRNA 5' to 3'. Now there are 20 different amino acids, but only 4 nucleotides, so we can't have each amino acid specified by a single nucleotide. In fact, the nucleotides will be read in groups of 3, which are referred to as codons, where each non-overlapping group will specify the next amino acid to be added to the chain. We'll look at this process in more detail in the video on translation. This is the codon table, and it tells us which amino acid is specified by each codon. Let's work with an example codon of AUG. Now, just to be clear, we're reading these codons in the 5' to 3' direction in the order they were synthesized. This table is sometimes formatted in a circle like this, and sometimes in a more conventional square or rectangular table, but the information is always the same. With a circular one like this, we're going to start in the middle and work outwards. Since our 5' base is A, that puts us in this section of the table. Moving out to the next ring, we see that our next base is U, so that puts us in this section. We then move out to the outer ring for the 3' base, which in this example is G. So, if we have a codon of AUG, this will give us the amino acid methionine, which is also the start codon for translation. Once the ribosome locates the first AUG codon in the sequence, that will then determine the reading frame or groupings for the rest of the codons. The ribosome will then continue to read the codons in a non-overlapping manner until it reaches one of the 3 stop codons, shown here in orange. There are a couple of key points that we can note about the genetic code here. First, we can say that the genetic code is redundant. We have a look at this section here. We can see that a codon of CC anything will specify the amino acid proline. In fact, a quick look around the circle will show you that a similar scenario is true of every amino acid, except for methionine. And in most cases, it's the 3rd base of the codon that is more variable. Now, this is what we refer to as wobble, where the 3rd base can be changed without affecting the amino acid sequence. We can also see that the genetic code is not ambiguous. The amino acid specified by a given codon doesn't change. AUG, for example, will always give us the amino acid methionine. UGC will always give us cysteine, and so on. The genetic code, as shown here, is also considered to be universal, with the exception of a very small number of cases. Codon sequences will be translated into the same polypeptide sequence, regardless of the organism the gene originated in. This means that a gene from one organism can be placed into another, and assuming it's successfully transcribed, it will be translated into the same protein with the same function, even though that gene doesn't normally exist in that organism. And this is how we're able to make transgenic organisms for various purposes, like agricultural or pharmaceutical applications. Thanks for watching, and please be sure to check out the videos with more details on the processes of transcription and translation.